Student, welcome to today's lesson. This is biology lesson for grade 12. In our today's lesson, we will see the consecutive phase of meiosis, which is meiosis II. Student, at the end of this lesson, you will be able to differentiate meiosis II from meiosis I. You will describe all phases of meiosis II. Meiosis II. When we say meiosis II, it is a second phase of meiosis cell division that evolved with the separation of sister chromatids. When we say meiosis I, it evolves with the separation of homologous chromosome. But here, we are going to speak about the separation of sister chromatid. And again, the activity that can be takes place during meiosis II can be depend on activity that can be takes place during meiosis I. The cell formed at the end of meiosis I is all haploid because it contains one set of chromosomes in homologous pair. This n number that can be produced at the end of telophase can be entered for short period of time because it is a ready one for next phase. Then after, the activity continue to meiosis II. By that, the activity separation of homologous pair that takes place during meiosis I is very important for the activity that can be takes place during meiosis II. As a result, the number of daughter cells that is formed at the end of meiosis II can be reduce the number of parental chromosome by half. Here, meiosis II can be differentiated or separated from meiosis I by the following main activity. There is no crossover during meiosis II of prophase II. The only crossover when homologous pair become exchanged, the chromosomal segment at chiasmata point can be takes place only during meiosis I, prophase I. No crossover during prophase II. And again, chromatid becomes separated during meiosis II as process of separation of sister chromatid. And again, as the process of separation of centromere, there is a separation of sister chromatid. And again, chromosome become line up side by side during metaphase II. It is a random alignment of chromosome takes place during meiosis II at metaphase II. By that, meiosis II can be considered as an equational division as it divides a sister chromatid, whereas meiosis I is considered as a reductive division as it is considered as a division of or separation of homologous chromosome. This is a minor difference that can be found between meiosis I and the meiosis II. But both meiosis I and the meiosis II can be takes place in an organism that can be joined a sex cell, a gamete producing cell, where both meiosis I and the meiosis II can have the same stage, prophase, metaphase, and anaphase, as well telophase. But there is different activities that can be takes place during meiosis I and the meiosis II. Stages of meiosis II. Meiosis II has the same stage with meiosis I. The same four stages found here too. Prophase II, and again metaphase II, anaphase II, and again telophase II. This each stage can be considered as meiosis II. Before meiosis cell division began, the original chromosome must be copied that can be held together by centromere during meiosis I. But here, there is no more interface. Means there is no copy. Because meiosis II can be begun from telophase II. The daughter cell that produced during telophase I become progress to that of prophase II. And there is no copy or duplication here. No interface. And again, during prophase II, chromosome become condense. Nuclear membrane disappear or dissolve. A spindle fiber that can move chromosome from one place to another through the cell can be reformed, that attached together still by centromere. Still, there is no division or separation of centromere. Centromere held together the cells that are produced from telophase I and pass to the next stage of meiosis II, and to prophase II, holding them together. There is no separation of centromere, that they can be having the original and the copy chromosome together. So this is the activity that can be takes place during prophase II. Another phase of meiosis II is metaphase II. During metaphase II, homologous pair, since original and the copy found together, because there is no separation of centromere still, homologous pair become aligned to the middle line of the cell. A spindle fiber brings to that of the middle cell at equilateral line of the cell here. They are arranged as a middle line here. They are not as a pole side. And again, centromere held them together. Centromere is very importantly joining the copy number with the original chromosome. This is what we call metaphase II. Another very important and critical stage of meiosis II is anaphase II. During anaphase II, one thing that you must be understand is there is a separation of sister chromatid, and sister chromatid become moved to opposite pole. Here there is a separation or division of centromere. We are not come to speak no more about homologous chromosome after anaphase II. We mean that during all stage of meiosis I and during some stage of meiosis II, like prophase II and metaphase II, we are speaking about homologous pair because original and the copy chromosome number found together. But here we cannot speak about centromere and the homologous pair here. Centromere division is found during anaphase II of meiosis II. Here, one that can move to the opposite pole here is the only sister chromatid. The copy number move in one direction, the original number in another dimension, and again they can move or separate from each other. Separation of sister chromatid is found here because there is no centromere here. There is division of centromere. The only stage of centromere division is found during anaphase II. What can progress to that of the next stage of meiosis II is telophase II. During telophase II, chromosome become decondensed. Nuclear membrane that disappeared during prophase II come to reappear here. A spindle fiber that is formed during prophase II that is moving chromosome through the cell come to disappear here. Because for daughter cells come to form it, there is a division of cytoplasm again, which we call cytokinesis. Finally, at the end of telophase II of meiosis II, 4 haploid daughter cell can be produced, where these 4 haploid cell can have half number of parental chromosome. Reduce the parental chromosome by half. If the parental chromosome is 8, the daughter cell that can be produced at the end of telophase II can be reduced the parental chromosome by half, which means 4. For example here, there is separation of sister chromatid here. There is no more homologous chromosome here because there is separation of centromere here. There is 4 daughter cell. 1 daughter cell, 2nd daughter cell, 3rd daughter cell, 4th daughter cell. The end product of meiosis at the end of telophase 2 is 4 daughter cell, where each daughter cell can be reduced the parental chromosome by half, and each daughter cell can be show a genetic variation because of crossover, because they can exchange their genetic segment that can be takes place during prophase 1 of meiosis 1. None of the daughter cell formed at the end of telophase 2 is identical. This brings genetic variation. Student, when you speak about meiosis, it evolves about spermatogenesis and the oogenesis. If the cell with 4 number of chromosomes wanted to be divided meiotically, at the end there is a formation of 4 daughter cells, where each daughter cell can be reduced the parental chromosome, 4 parental chromosome, and again the one produced at the end can be reducing the parental chromosome. There is 4 daughter cell, where each daughter cell can be half number of parental chromosome. So at the end of meiosis, we can end up with 4 daughter cell, where the parental chromosome can be reduced by half. This is an example of spermatogenesis, which takes place in male gamete producing. And again, oogenesis can be follows the same strategy in the mechanism again. A student, generally, meiotic cell division is very important to generate a genetic diversity through a living organism. This is as a result of crossover, which is important to exchange a genetic segment between homologous pair of an organism during meiosis 1 at prophase 1. As a result of crossover, none of the daughter cells formed at the end of telophase 2 are identical. They can reduce the parental chromosome. And this is very important in creation of unique offspring, a variable offspring across a generation. One that can be determined an organism genetic variability is the short section of DNA, the one that we call gene. Gene is very important in determining an organism traits and variation. There is one honorable biologist called Thomas Hart Morgan that carry out a research over understanding and proving a gene which is a section of DNA that can determine an organism trait or feature, which is a basic for physical inheritance. He carried out a research on fruit fly. He experimented on this animal to understand, as a result of crossover and the recombination, there may be a genetic variability among an organism over a generation. He preferred this animal, which is a good experimental animal, in ensuring genetic variability over a generation, because this animal is very small enough with short life cycle. And again, it is very cheap. And again, this animal can have 4 pair of chromosomes, large and easy to observe. By that, he proved and got a Nobel Prize over the crossover recombination, and the gene found over the chromosome is a basis for physical patterns of inheritance that can determine a unique offspring production over a generation. Student, in our today's lesson, we have seen as meiosis II is an equidistant division that can evolve in separation of sister chromatid. At the end of meiosis II, there is a production of 4 daughter cells that can have half number of a parental chromosome. And again, as a result of crossover, exchange of a genetic segment, none of the daughter cells is genetically identical. And again, homologous chromosomes become aligned together as the equilateral plane during metaphase 2. And the separation of centromere can be takes place during anaphase 2 that move to opposite pole. This brings us all about the biology lesson for grade 12. Goodbye, student.